All right, we've come a long way. We've talked about the basic components and the structure of the cell surface, the plasma membrane. We've talked about how stuff moves in and out of cells. We've talked about how cells use vesicles in the cytoskeleton to capture stuff or to send stuff out of the cell, right? Now let's talk about some unique features of cell surface, some specializations of that cell surface. So we talked about the extracellular matrix, okay? So again, remember that extracellular matrix can look different depending on what type of cell it is, okay? Most animal cells are going to have collagen and cadherin proteins on the outside of that extracellular matrix, which let that cell attach to other cells, okay? Which keep all of our cells in a very specific order and organization. Integrin proteins are proteins that span the plasma membrane and basically connect that extracellular matrix, those collagen fibers and those cadherin proteins, to the cytoskeleton on the inside. So that way, if there's damage to that extracellular matrix, if something changes in that extracellular area, that information can be transmitted via the cytoskeleton, which can eventually activate other proteins, which can change the signaling pathways going on in the cell. Okay? So cells can kind of sense their environment and feel what's going on around them, I guess you could say, with their extracellular matrix connected to their cytoskeleton via the integrin proteins. So now we're going to talk about some special, unique types of extracellular matrix connections between cells. So how cells stay connected to each other, some unique special cell junctions. So we find these in animal cells. Okay, there's a whole other set of cell junctions in plant cells, but we're going to focus here on animal cells. So the first type of cell junction that I'm going to talk about are tight junctions, and just like the name implies, they are tight. They are basically where the plasma membrane is held together by continuous connections of proteins between two cells. Okay, so it creates a continuous watertight seal. Okay, so tight junctions are watertight. Okay, so there's proteins that connect on either side of that plasma membrane that hold that plasma membrane, hold those two cells very, 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 very tightly together. So that way water cannot escape, ions cannot escape. Okay, tight junctions are very common in epithelial tissue. In places where we don't want water going in and out, those cells will be connected together by tight junctions. So think like our bladder, right? We want that bladder to be watertight. We don't want that urine, a waste product, to go out into the rest of the body. Our skin, right? Our epithelial tissue on our skin, our dermal tissue, that's often tight junctions to prevent water leaking, so we're not just like oozing extracellular fluid all of the time. Desmosomes are a type of junction that are flexible and hold cells together, but not super tight. Okay, so desmosomes let water and ions flow through, okay, between the cells, but they still keep cells anchored to each other, but there's a little room for flexibility. So we find these, I like to think of these kind of like stitching in a quilt, right? You take the two pieces of your quilt, but there's going to be stitches throughout the quilt that keep those pieces of the quilt all together, but they're not super, super tight, right? There's a little bit of flexibility. Or kind of like a button being connected to some pants, right? You don't want that button to be right tight up against the fabric of the pants because you want to get the loop around it, right? So desmosomes allow for some flexibility for those cells to move against each other, okay? And they're anchored in the cytoskeleton by keratin proteins. Okay, so keratin proteins will connect to the other cytoskeleton microfilaments and fibers and anchor those cells together. Okay? But they're still connected, but they're further apart and they're flexible. So these are really important, desmosomes are really important junctions in tissues that stretch. Okay, so like skeletal muscle, stretching and contracting. Our skin needs to stretch and contract, so those cells need to be able to move against each other but while remaining attached to each other. So desmosomes allow that to happen. Okay? If you ever pull a muscle, right, If you ever pulled a muscle and you're like, oh my god, that really hurt, that muscle's not really feeling like it's working like it's properly working, that's probably because you actually ruptured some of those desmosomes. So our muscles kind of come in sheets of cells, and if any of those sheets, those desmosomes, are ruptured, you pulled that muscle. Gap junctions are continuous connections between the cytoplasm of two cells. So they are basically little tunnels that connect two cells together, or maybe multiple cells together, right? So these little tunnels let the cytoplasm between the two cells exchange. Okay, so water can flow these gap junctions, ions can flow through these gap junctions, sugars, amino acids, small molecules can pass through these gap junctions. Okay, these are important in cells that need to be tightly coordinated in their functions. So like the cells in our heart muscle are connected, our cardiac cells are connected by gap junctions because we need all of those cells to send an action potential at the same time. We don't want cells in our left ventricle not working at the same time or the same pace as the ones in our right ventricle, right? We want them all in sync, working together, and so gap junctions help keep that coordination of our heartbeat going. Okay. So gap junctions connect cytoplasms together. Desmosomes are a flexible connection anchored with keratin proteins in the cytoplasm. Tight junctions create a watertight barrier to help create basically a watertight barrier between cells and the rest of the body. Think like our bladder or outside tissue, right? And then the type of cell surface specialization is going to be microvilli. Okay, so this is not a connection between cells. This is an extension or kind of a folding of that plasma membrane to increase the surface area. Okay, surface area is a huge deal when it comes to cells. Cells are small in order to maximize their surface area. Microvilli help to maximize the surface area of that plasma membrane, right? Why would they need to maximize the surface area of their plasma membrane? Well, everything that cell needs to bring in or out of the cytoplasm has to go through that plasma membrane. The more surface area you have of the plasma membrane, the more efficiently you can move materials into or out of your cell. We find microvilli in human cells covering our small intestine. Our small intestine is absorbing a lot of the nutrients from our food that we eat. Everything that's getting broken down in our stomach and continuing to get broken down in our intestines is going to be transported into the intestinal cells and then into our circulatory system. So that way we can feed the rest of our body. So in order to do this efficiently, in order to maximize the amount of absorption that occurs in our small intestines has these microvilli. So these are different than cilia. Cilia are connected to the cytoskeleton so they can move. Microvilli are not. They are just folds of that plasma membrane. They are not cilia. They're not actively moved back and forth. Okay, they're microvilli. They look kind of the same, right? They kind of have a hairy texture, but they are not connected to the cytoskeleton. They do not move, so they're not cilia. And this is also random, but this is why if you ever feel the inside of an intestine of a mammal, like if you dissect an animal and you feel the inside of the intestine, it feels like velvet, very, very soft. This is why, because of these microvilli. Okay, microvilli increase the surface area. Surface area is the name of the game when it comes to cells.